Thank you, Alex, for giving me this opportunity and to incorporate me in one of the resource persons. And I'm very much happy and to see that in India we are taking such initiatives where we all people from academia, industry, and also from regulatory bodies coming together to share our knowledge with each other. That's really a very good initiative, and Alex has taken that initiative. And we can see now India will become one of the, you can say, in the coming day, where we can see our toxicologists around the world. So with this, I would like to talk about the introduction part. Now we'll discuss about the classifications of this natural toxins, animal toxins, and the most important part is the mechanism, how these plant-derived toxins lead to the toxicity, in the same way like how these animal-derived toxins lead to the toxicity. We'll do some case study and we'll also discuss the routes of the exposures and toxicokinetics of these toxins from plants and animals. What are their various clinical signs, treatment approaches, epidemiology. I'll discuss a few aspects of these about plant and animal toxins. And the most important is the prevention and safety measures. And I would like to summarize my presentations with some takeaways and future toxicology in this particular area. So coming to the introduction part, so this session is focused on the toxins that is produced by the plants and animals, and these substances can affect the human health through either maybe through the accidental ingestions or maybe in venomation. So in venomation, like it's a process where some venom through this insect bite is entered into our body, or maybe due to this environmental exposures. So these natural toxins are of great importance, not only for their potential harm, but we can say they can also having been contributed in the science. And many were the first natural experiments where these natural toxins help in the foundation of toxicology. For example, like snake venoms for to poisonous plants like oleander, and we have to understand these toxins to develop various antidotes. And we can also help in the prevention measures and how we can do the safe handling practices of these toxins. Now coming to the historical perspective, so these are natural poisons which used in ancient medicines and warfare. If you see this, this indigenous communities, we also use these plant extract and animal venoms in hunting and warfare. So before, so they are not like recently been discovered or investigated. Since the civilization happens, we used in for one or other purpose. If you can see this curated poisons in the dart for hunting of the animals. We also have some historical cases of the deliberated poisonings, and I think you all have heard and read about this Socrates scripts, where this hemlock ingestion done for this poisoning, and we also know how the societies were aware about these dangers. So over the centuries, our understanding of these toxins evolved from folklore to the scientific analysis, and now we have lined the groundwork of modern toxicology. Public health significance. So plant and animal toxins still possess the major public health concern. Each year, thousands of the cases of this accidental poisonings occur, particularly in children's and livestock. In veterinary importance, if we can discuss it, so veterinary medicines must also address this plant poisoning in this grazing animals. through which these poisons enter into the animal's body and lead to various toxicological responses. Further, if we talk about this environmental and agriculture implications, so these natural toxins can contaminate food supplies. For example, the cyanogenetic glycosides, a very popular example. It's come into when we improperly process this cassava. So they may lead to the accidental, this contamination of the cyanogenetic glycosides, which is very toxic to the body. So recognizing these risks is very, very crucial for this developing the preventive strategies and policies. Now, coming to the most important part, that is the classification of these natural toxins. So we classify this into two parts, either based on their chemical structure. So we have a vast chemical classes which they have some kind of the homology and similarity, or we can also classify these is based on the sources. But based on the sources is more classical one, where we classify these plant toxins into two major group, that is plant toxins, and they include like alkaloids, glycosides, and other protein-based toxins like lectins. And on the other side, we have animal-derived toxins, where we have venoms from the snakes, scorpions, and these toxic substances from the marine species, which are of important, as well as like this biogenic amines, such as histamine in the spoiled fish. So these dual classification help us to understand what their biological origin as well as their mechanism of action. So I'll discuss one by one these plant toxins. I'll cover some of these plant toxins, most popular plant toxins, then their mechanism of toxicity, and in the similar way, animal toxins and their toxicities. So coming to this plant-based toxins, so number one class here is the alkaloids. I think most of you have seen these type of toxins in the laboratory as well. So alkaloids is nothing. These are the biogenic amines containing nitrogen, and they are mostly affect the nervous system and the cardiovascular systems. These are the most two prominent effects, or you can say the toxicological effect of these alkaloids. For example, nicotine act as a nicotinic acetylcholine choline receptors, while atropine blocks the muscarinic receptors. And leading to this anticholinergic systems, toxins found like in yew plant, and it is particularly dangerous due to its potent cardiotoxic effects. So these substances illustrate the wide range of this biological actions of this alkaloid on both human and animals. Coming to the next class, glycosides. These are another class found in the plants, and basically they have a sugar moiety, glycone and aglycone moiety. So glycone is basically a sugar moiety, and aglycone is the active compounds. So if it's a glycoside is toxic, we usually refer this as a active toxic compounds. They are of various, this chemical natures. These glycosides basically having a cardioglycoside that is found in the foxglove leaves. Like we also told, like told this foxglove as a digitalis leaves. So they have a digoxin chemical moiety, digoxin. So it's act on sodium potassium ATPase pump, which may lead to the dangerous antiarrhythmias. So this is very, very toxic and fatal side effects. And this also like cyanogenetic glycosides, which is found in cassava and cherry pits. So which release cyanides upon metabolism and may lead to the impairing into the cellular respirations by blocking the cytochrome oxidase. So both these poses a significant risk to the humans and animal health. Now coming to the specific class of this glycoside, that is cardiac glycosides, that is found in digoxin, which is found in the foxglove, and oriandin from this oleander. I'll also produce one case study on this oleander, and they are well known for their potent cardiotoxicity. So they basically inhibit the sodium potassium ATPase pump in the myocardial cells, which increase the intracellular calcium levels and enhancing the contractility, but also predisposing to the heart arrhythmias. So digitalis derivatives are also used therapeutically because they have that kind of therapeutic effects we have explored through this toxic effects of this digitalis. And this plant exposures can cause vomiting, bradycardia, that is AV blockage, and potentially fatal for the fatal arrhythmias. Now coming to the another class, that is the cyanogenic glycosides. So these cyanogenic glycosides basically this liberate the hydrogen cyanide when metabolized in the gastrointestinal tract. Cassava is a major dietary source that requires proper preparation to avoid the toxicity. So cyanide binds to basically to the cytochrome oxidase in the mitochondria and may lead to the oxidative phosphorylation. So this prevents the ATP production and leading to this cellular hypoxia, which despite adequate oxygen delivery. So clinical symptoms include basically headache, confusion, seizures, and rapid cardiovascular collapse with the cyanogenetic glycosides. Now coming to the protein likes, or the another class which we usually refer as lectins and toxalbumins. So these are carbohydrate binding proteins basically, and they can disrupt the cell membrane and intracellular functions by binding the intracellular proteins. Toxalbumin such as ricin from castor beans and abrin from rosemary peas. So these are among the most lethal plant toxins known, and they act by inhibiting the protein synthesis at the ribosomal levels, and they may lead to the cell death. Just a few milligram can be the fatal, and making them of both medical and bioterrorism concern. So this is they have a very less therapeutic window, and a very small dose may lead to severe toxicity both in humans and animals. Now, other plant toxins. So we have discussed like these major toxins groups. So several other plant toxins are also important, like pyrrolizidine alkaloids, and these basically found in the plants like ragwort, which may lead to hepatic metabolism. When it's go to the hepatic metabolism, it lead to the toxic intermediates. After metabolism, it leads to the toxic intermediate that can cause veno-occlusive disease and cirrhosis. So the certain plants, such like kind of this poison ivy release urshol, causing severe contact dermatitis via the delayed hypersensitivity reactions. So through some contact, there are certain plant species known when it's come in contact with the humans and animals, they may lead to various dermatitis, which may even lead to the fetal and severe organ failures. So these examples basically highlights the diverse organ targeted by these plant toxins, from skin to liver, and also this GI tract. Now, if we talk about the mechanism of action of these plant toxins, so these alkaloids, basically this nicotinic alkaloids, for example, this nicotine, quinine from poison hemlock. So they act as an agonist at the nicotinic acetylcholine receptors, and may lead to the overstimulation of this acetylcholines, and which lead to the depolarization block and lead to the paralysis and respiratory failures. Whereas this tropin alkaloids, which is found in this atropine, which is found in this tropin, in the Datura plants, basically having an active ingredients like atropine and scopolamine. So this basically block the muscarinic acetylcholine receptors. So they show the anticholinergic syndromes, may lead to dry mouth, dilated pupils, hallucination, and tachycardia. We also use therapeutically, but these high doses may lead to these side effects. But we also having some therapeutic applications of these tropin alkaloids. So we use basically in conjugation with this anesthetic agent, because this anesthetic agent may lead to hyper secretions. So sometimes we required as a therapeutically, but accidentally, you can say the consumption of this tropin alkaloid or high dose may lead to these type of toxicities. Coming to this indole alkaloids, it's found in the nux vomica, strychnine and brucine. So they are highly toxic, and they lead to the blockage of glycine receptor in the spinal cord, and they may lead to the loss of inhibitory neurotransmissions, and may lead to the severe conversions, both in animals and humans. So this is the mechanism of actions I have summarized in a very flow chart way, so that we can understand the sequences. Now coming to these glycosides, I have already discussed this cardiac glycosides, for example, digoxin and this alendrine, which is found from this ornamental plant. So basically, what they do, they inhibit the sodium potassium ATPase pump. So it increases the intracellular sodium, may lead to the increased in intracellular calcium deposition. So there is a strong cardiac contractions, and may also having the risk of arrhythmias. Arrhythmias basically it's irregular heartbeat, or there is incoordination between PQRS waves. So that's a very potential side effects of this cardio active glycosides. These cyanogenetic glycosides, amygdalin in apricot kernels, or it is also found in almonds or cassava. So it's released basically hydrogen cyanide, and it inhibits the cytochrome oxidase in mitochondria, or which blocks this oxidative phosphorylation, may lead to this cellular hypoxia. And this cellular hypoxia, despite the normal oxygen levels, and may lead to the cell death. The other class, this lectins and protein toxins. So what they do, they this is resin from the castor beans. So it inactivates these ribosomes by deactivating 20S RNAs, which lead to the complete shutdown of protein synthesis, and may lead to the mm, this cell death. And this abrin from Jacquerie P. So the mechanism is very much similar with resin, but it is more potent. Now coming to this terpenes and other compounds. So texines from yew plant. So it's block sodium and calcium channel, may lead to this cardiac conduction block and arrhythmias. And this pyrrolizidine alkaloids, it's usually metabolized in the liver to toxic intermediates. That intermediates bind with DNA and proteins, and may lead to the hepatotoxicity and cancer risk. Now coming to the other class, that is animal venoms. So the general features of these animal venoms, so they are not the single toxins, but they are the complex cocktails of the proteins, peptides, and enzymes. So they are evolved for prediction of defense, and often acts very rapidly as compared to the plant toxins. And depending upon the species, Venoms may target the nervous system, cardiovascular system, or they may lead to this blood coagulation pathway. So these are few more, you can say, the important targets of these animal venoms. That is nervous system, cardiovascular system, or this blood coagulation pathway. And their potencies and rapid actions make them particularly dangerous and medically significant. Now coming to discuss one by one the most important these animal venoms. So first one, of course, the snake venoms. So it is broadly divided into neurotoxics, which affects the nerve transmission, as we found in the cobras, and hemotoxic, which basically damage the blood and tissue, as most common in the vipers. So many venoms also contain these proteolytic enzymes that may lead to the tissue necrosis and systemic clotting abnormalities. So the clinical patients may present with paralysis, severe bleeding, shock, or may have some kind of localized tissue destruction. Antivenom remains the primary treatment, but supportive care is often crucial in case of this venom toxicities. Coming to the spider venoms, so these spider venoms basically they are very in action. So the black widow uh, spider, that's also having a compound like alpha letrotoxin, which basically cause the massive neurotransmitter release, may lead to the severe muscle cramps and automatic disturbances. In contrast, this brown recules, that is Loxosceles venum, which contain the sphygmomyelinase D. So this sphygmomyelinase D is basically the compound toxic venom, which trigger the local tissue necrosis, hemolysis, and systemic effects in the severe cases. So while fatalities are rare, the mobility from the bites can be the significance as compared with the snake venoms. Uh, Another subclass in animal venoms is the scorpion venoms. So what this scorpion venoms do? So they affect the sodium and potassium channels, and these are medically important in many of the tropical regions in the world. So they're disrupting the normal nerve and muscular functions. Patient often present with severe pain, excessive salivation, and sweating. Trachycardia is one of the important clinical representation, and hypertension. And in children and in adults, this may progress to arrhythmias and pulmonary edema. May lead to shock. So the supportive care and when available, that antivenom can be the life-saving medication in the case of this scorpion venoms. Coming to the next subclass of this animal venom, we have amphibian toxins. So this amphibian toxins, this has been secreted by many amphibians through their skin as a defense mechanism. So torts, for instance, produce buffer toxins that resembles with the cardiac glycosides that we have seen, like digitalis. We have two class, this cardinolides and bufferinolides. Cardinolide basically present in digitoxins, and this bufferinolides present in this salimarin is a drug, salimarin, and this toad. Uh, so they have a similar chemical structures, salimarin and bufotoxins. So they're leading to the digitalis like poisoning symptoms, they have having they having arrhythmias and hallucinations. So indigenous culture have the historical used in this traditional medicines in rituals or as a arrow poisoning. And this modern exposures occur from accidental ingestion or misuse of so-called toad licking. So they may lead to severe toxicity in the humans and animals. Marine toxins is the other subclass. So these marine toxins are very much diverse and often linked with the algal and fishes, mm, blooms. This shellfish, which can accumulate toxins such as toxin, leading to paralytic shellfish poisoning. Tetrodotoxins, famously found in puffish, it blocks the voltage-gated sodium channels and prevent the action potential propagation and causing the paralysis. So basically, it acts on this voltage-gated sodium channel, which is present in the neurons, and may often lead to the respiratory failures once a person consume these toxins. Since these toxins are heat stable, cooking does not neutralize them. So though they are the proteins, but it's heat stable proteins, they may lead to the toxic effects. Cone snail toxins. So this is another subclass of animal toxins. These corn snail produces conotoxins, and it is a family of a small peptide that selectively block the ion channels and receptors. Basically, they block the calcium ion channels, and they are capable of causing the rapid paralysis and death in the humans. Usually, though respiratory arrest may happen, but these peptides are also being studied as a template for novel analgesic and therapeutic drugs due to their high selectivity for the neuronal targets. So as I've already told, so these toxins has given us the foundation in modern toxicology to discover the new drugs from these toxins, or we can repurpose this for other disease. Now, very interestingly, there is a bacterial symbiosis and toxic production. So, interestingly, many marine and amphibian toxins are not produced by these animals themselves, but by the symbiotic bacteria. So, tetrodotoxin, for example, is found in puffish and some octopus, but its true source appear to be the marine bacteria living within these hosts. So, this symbiosis complicate this ecological understanding and may lead to influences toxins variability across the various species and regions. Now, comparative potency of animal toxins. So we have discussed a few animal toxins here, like snake, venom, spider, scorpion, marine from the marine sources. So these, like if we compare their potency, so these animal toxins are among the most potent substance known often for this surpassing the synthetic poisons. For instance, botulinum toxins and tetrodotoxins are lethal in nanogram doses. So small dose can be lethal and may lead to the toxic side effects. And their remarkable potency has made them invaluable research tools in neurobiology, as I've already told that we are targeting these toxins for as a pain as a potent analgesic for the discovery of painkillers. So they may lead to the unrevealed synaptic transmissions and ion channel functions. So this same potency also make the accidental or intentional exposures may be highly dangerous. Now, likewise, coming to the, I have already discussed like the mechanism of these uh, mm, uh, plant toxins. So, like we are now going to summarize the mechanism of this animal toxins. So, we talk about this snake venom. So, it's basically the neurotoxic like in cobras and krates. So they have alpha neurotoxins bind postsynaptic nicotinic acetylcholine acetylcholine receptors, may lead to the paralysis. And if it is bind to beta neurotoxins, which inhibit the parasynaptic acetylcholine release, may lead to the flaccid paralysis. Hemotoxic venoms, which is present in vipers, rattlesnakes, they contain the protease and the phospholipase, which cause the hemorrhages, coagulopathy, and tissue necrosis. Talking about this cardiotoxic venoms, for example, some cobra species, so they directly damage the cardiac muscles and having the arrhythmias and cardiac arrest. Spider venom and scorpion venoms. So if we talk about this black widow spider, laterotoxins, they have laterotoxins. So they forms the pore in the presynaptic nerve terminals. So they lead to the massive neurotransmitter release or leakage in the parasynaptic nerve terminals, having the painful muscle spasm and autonomic dysfunctions. This brown recurse spider, this sphygmomyelinase D, they have the compound sphygmomyelinase D. So it destroys the cell membrane and having the local necrosis and hemolysis. These scorpion toxins, basically, it's prolonged the sodium channel opening. So sustained depolarization may lead to the autonomic storms like hypertension, arrhythmia, sweating, salivation, etc. This marine toxins, tetrodotoxins, or which is found in the puffer fish, it block voltage-gated sodium channels and prevent the nerve conduction, paralysis, and respiratory arrest or blockage. Saxitocin, which is found in the red tide shellfish poisoning. So its mechanism is similar as in the tetrodotoxins, and it lead to block the specific sodium channel ions. Sugotoxins, which is found in the reef fish. So this opens the sodium channel. Persistent depolarization may lead to GI, gastrointestinal upsets, neurological symptoms, or maybe hot cold reversible. So they also affect the central nervous system. And there is a domic acid, that is a algal toxins found in the algae. So it is excitotoxins, basically overstimulate the glutamate receptors, which may lead to the neuronal injury, seizures, and memory loss. These are pod venoms also. They are like bees, wasp, and ants. So basically, they contain the meltin, phospholipase A2, histamine, and hyaluronidase. So it's basically have a local effects that may lead to the local pain, inflammation, and sometimes they may lead to the systemic anaphylaxis, like when it's been done in the bulk or when there is more stings and more bitings by the bees and wasps, so may lead to the anaphylactic shock as well. There's a multiple organ failure at a one time. So now coming to concluding these two classes, that is plant and animals. So the key insights these both plant and animal toxins, they produce the toxic compounds. Their route of exposure differ, but these plant toxins are usually ingested, leading to the delayed systemic effect, and such as liver failure or cardiac arrhythmias. In contrast, these animal toxins, they are directly, through biting, they are directly coming to the systemic circulations, and they are bypassing the natural barrier and often causing the immediate life-threatening effect. So, animal poisons are more dangerous as compared to this plant toxins, and both categories can result in the severe illness or death, and may underscores the importance of this rapid recognitions and treatment help the humans and animals. Now, the routes of the exposures of these toxins. So, these natural toxins, they enter the body through three primary routes, that is ingestion, injection, and dermal contact. Plant poisons like cardiac glycosides or these cyanides are typically ingested. While these venoms from snakes, scorpions, or spiders are basically delivered through this bite in a form of like injections, and some plant toxins like earth from this poison ivy cause skin irritant upon contact. So understanding the route of exposure is very, very critical for this clinical diagnosis and for this emergency management of these toxins. Now, coming to the diagnosis of this natural toxin poisoning. So, this natural toxin poisonings basically relies on the patient history and characteristic of this clinical feature, such as maybe the laboratory assay for specific toxins are very, very limited. For example, a child presenting with bradycardia and vomiting after ingestion oleander leaves suggest cardiac glycoside poisonings. So clinicians must also have distinguished these syndromes from other cause of arrhythmias, seizures, or skin lesions, making through the historical backgrounds or maybe through the experiences or their practices. Now coming to this clinical management and principle. So once there is a poisoning, either through plant or through this animals, so the most case of require these supportive cares like airway management, fluid replacements or fluid additionals, and symptoms treatment, where we specific antidote like digoxin specific fragments we have for cardiac glycosides toxicities, and antivenom for snake bite should be administered. So these are the specific clinical management, and now we also decontamination measurements such as activated charcoal or may require the gastric leverage, which may be appropriate only in the selective ingestion and when performed early. So they may help for delaying the absorption or stopping the absorption of this ingested poisons from plants. Now, antivenoms. So, antivenoms are basically these antibodies which is prepared and that bind and neutralize the venoms toxins, and this prevent them from acting on the tissues. So they are highly effective, but must be administered promptly, ideally before they will lead to this irreversible damages. However, these antivenoms may carry risk of allergic reactions in certain individuals, or may lead to the kind of a serum sickness, or may lead to the overloading of antibodies in the serum, and their availabilities may be varies globally, making them critical. But sometimes it's a limited tool in the toxic managements of this type of poisoning. Now, I would like to discuss few case studies. So first is this oleander poisoning. So oleander is a very common ornamental plants, and it contain a potent cardiac glycoside. Ingestion, even a small amount of leaves or flowers can cause the nausea, vomiting, bradycardia, and life-threatening arrhythmias. Diagnosis is basically based on the clinical symptoms, which is supported by the ECG findings. Treatment involves the supportive care, like digoxin fabs, or there's a fragments if available. And this can underscores how common plant can be the lethal risk. Now, the snake bite. So viper bites are very common in rural areas, often present with swelling, pain, and bleeding at the bite site. Systematically, patient may develop coagulopathy, hematuria, and shock due to hemotoxic effects of these venoms, and they having this prompt administrations of this correct antivenom is critical. So local wound care, tetanus prophylaxis, and management of secondary infections are also essential component during the treatment of this snake bite. Another very important and interesting is the puffish ingestion. So puffish serve as a fugu in Japan, which contain this tetrodotoxin, a potent sodium channel blocker. So symptoms develop like rapidly, beginning with this numbness and tingling, followed by paralysis and respiratory failure. And there is no antidote available. So survival depend on rapid initiation of this like kind of mechanical ventilation until the toxin is metabolized. Despite it risk, fugu remains a delicacy, highlighting the cultural dimensions of the toxic exposures. In basically in Japan, it's very common, and there is a specific cook, like a very high salary paid, where they have the idea how this fish has to be prepared and served to the customers. Now coming to the therapeutic uses of this natural toxins. So I have talked about this toxic effect, but there is a two like there is a two another part as well. So interestingly, these natural toxins have very having very important therapeutic applications when used in controlled dose. For example, like digitalis, it is derived from this foxglove, remain the treatment for heart failure. And this botulinum toxins, one of the deadliest substance known is widely used for the muscles spasticity and in cosmetic procedures. Uh, and there is gluconotide, a synthetic peptide based on the cone snail venom, is used for severe chronic pain. So this demonstrate how poisons can be transformed into the medicines. Now, role in toxicology research of these plant and animal toxins. So these natural toxins have serve as a vital tool in the toxicology and pharmacology research. Venoms like tetrodotoxin and ponotoxins have been used to study the sodium and calcium channel respectively. So we use to create a model in animals, and they're the very useful models for understanding the sodium and calcium ion channels. Also, major advances in neurophysiology of these toxins. So, plant toxins such as colchicine have helped in elucidate the cell divisions mechanism. We use it during our toxicity studies. And this chromosomal abbreviations studies. And these substances, while dangerous, but they have instrumental in deepening our scientific understandings in the biological system. Now, public health implications. So, plant and animal poisoning remain a serious public health issue worldwide, especially in the developing countries. In rural areas, snake bite, scorpion stings, and ingestion of these toxic plants are very frequent due to close human environment interaction. These exposures contribute significantly to the morbidity and mortality. However, the public health measures such as awareness campaigning, educational seminars and workshops, safe food preparation, and access to these antivenoms are very, very essential to reduce this burden. Epidemiology and poisoning. So, if we see this epidemiological data show that the snake bites affects hundreds of thousands annually, and leading the WHO to classify is as a neglected tropical disease. Children are disappropriately affected by the plant exposures, and often due to the accidental ingestions, these marine toxins cause seasonal outbreaks of shellfish poisoning in coastal communities. So, recognizing these epidemiological trends help the target preventive and treatment resources effectively. Now, case fatality and prognosis. So, the prognosis of these natural toxins depend on the several factors: the species involved, the dose of toxins, and the availability of treatment. Untreated snake bite in rural areas may carry case to the fatality rate exceeding 20%, while access to the antivenom reduces the mortality dramatically. And this plant poisoning ranges from mild GIT upset to the rapid death as a aconitin this ingestions. And early recognitions and interventions remain the best predictor of the survival. So coming to the summary part of my presentation. So natural toxins from plants and animals represent both a threat and an opportunity. They cause a significant illness and death worldwide, particularly in resource-limited regions. And at the same time, they have shaped the modern pharmacology and medicines, providing drugs, research tools, and medicines. They are also balancing the risk and benefit defines the role of toxicology in public health and science. So, concluding to this today presentation, the study of these natural toxins highlight their dual nature. Of course, they are deadly poisons in one context, whereas they are also life-saving medicines in the another context, raising awareness, improving prevention access to the treatment, and emphasizing the prevention can mitigate their dangers. Meanwhile, continuing scientific exploration while likely yield the new therapeutic breakthroughs. So this dual legacy ensure that plants and animals remain central part of the toxicology. Now, what's the future directions and what's the future of these plants and animal toxins? So, these future toxicology are now will harness this genomics and proteomics to this unreveal the complexity of these natural toxins at a molecular level. The synthetic biologists are now approaches may allow to scalable the production of antidotes and therapeutic toxins derivatives, and moreover the potential medical applications of these natural toxins from pain relief to cancer therapy are now expanding the bridge between the toxicology and the drug discovery. So with this, I would like to thank you for all the participants for listening patiently.